My name is Emily Balskus, and I'm a faculty member in the chemistry department at Harvard University. I am fascinated by microorganisms. Microbes like bacteria, archaea, and fungi are by far the most numerous organisms on the planet. Though scientists have traditionally studied microbes individually in the lab, in their natural habitats they live together in complex communities called microbiomes. Microbiomes are everywhere: the soil, the ocean, wherever we look we find them, including the human body. Each one of us is home to 5,000 times more microbes than there are people on Earth. Just like a fingerprint, each human microbiome is unique. No two people have the exact same combination of microbes. But what are these microbes actually doing in our bodies? Well, the answer is chemistry. Like all living organisms, microbes perform chemical reactions in order to survive. They consume nutrients from their surroundings, and they transform this food into the building blocks and energy they need to grow and reproduce. Microbes also make molecules in order to communicate with nearby organisms. Given how closely our lives are intertwined, we understand surprisingly little about the chemistry going on in the human microbiome. We don't know how these microbes interact with us, and we don't know how the chemistry they perform in and on our bodies is affecting our health. Research in my lab is addressing this problem. We are identifying chemical reactions from the human gut microbiome that are linked to health or disease. We're working to understand how these transformations actually happen, and we're trying to develop strategies to control this microbial chemistry. Many of the projects in my group focus on discovering enzymes, the protein-based catalysts that allow living organisms to perform chemical reactions. We find new enzymes by searching DNA sequences from microbes or microbiomes. Recently, my group has identified a microbial enzyme responsible for converting choline, an essential nutrient, into trimethylamine, a molecule that's responsible for the odor associated with rotting fish. This reaction occurs in the human gut, and an excess of trimethylamine production has been linked to heart, liver, and kidney diseases. Like much of the chemistry associated with the human microbiome, formation of trimethylamine is exclusively microbial. Human cells can't produce this molecule. Before our work, no one knew which microbes produced trimethylamine, so it was impossible to predict whether a given person's microbiome could perform this chemistry. Now, we can identify these microbes simply by looking to see if they have this key trimethylamine-producing enzyme, choline trimethylamine lyase. We're also studying the details of how this enzyme works. With this knowledge, we've begun to develop drug-like compounds that interfere with trimethylamine formation. The ability to control trimethylamine production may help us to understand its role in human disease, and could even provide a new strategy for treating or preventing these conditions. We could drug our own microbiome rather than our own cells. There's a tremendous amount of chemistry just waiting to be discovered in the human microbiome. My group studies how gut microbes transform compounds present in food, how they alter drugs we take, and how molecules they produce may influence the development of cancer. Understanding this microbial chemistry and its influence on human biology could have an enormous impact on medicine and nutrition. The research I do requires knowledge and techniques from different areas of science. I have the privilege of working with an amazing group of both chemists and biologists. Exploring and learning with them is truly the most rewarding part of my career, and I'm incredibly grateful for their hard work and their many contributions. Finally, we all thank the Dreyfus Foundation for their generous support of our work.